Imagine being in high school, looking forward to college, and the endless opportunities that await, only to have that dream crushed because of a devastating diagnosis of Ewing sarcoma, the second most common bone cancer in adolescents and young adults. Each year, about 200 children and teens in the United States are faced with this devastating diagnosis, with a very poor survival rate. Especially in patients that have widespread disease, the vast majority of these patients will die. And even those that survive suffer from significant toxicities of the therapies that we've given them, such as secondary cancer. There's an urgent need to identify novel therapies. My research project is looking into the tumor microenvironment of Ewing sarcoma. The tumor microenvironment is an intricate ecosystem that consists of cancer cells and non-cancer cells, such as immune cells. My research project is looking into a specific type of immune cell called macrophages in human Ewing sarcoma tumor samples, but also in a novel genetic model of Ewing sarcoma in zebrafish that's been recently developed by the Amatruda lab in hopes to identify novel treatment therapies. The tumor microenvironment plays a crucial role in Ewing sarcoma. Macrophages typically help us fight infection, but in Ewing sarcoma, it's known to make it harder to treat. Now, by understanding how these cells are organized spatially in the tumor microenvironment, this will allow us to understand how the interactions are occurring within the architecture of the tumor microenvironment in hopes to identify novel therapies and finding a cure. I'm using a new technology called Visium spatially resolved transcriptomic, which is the effort to understand the spatial organization of these immune cells and cancer cells. With this new technology, I'm able to see where specific genes are being turned on and off, and how the macrophages are organized around the cancer cells, and the interactions that are occurring between the macrophages and the cancer cells. I'm not only able to study this in human Ewing sarcoma tumor samples, but also able to generate the same tumors in zebrafish. Zebrafish share about 70% of the genome with humans and have very similar immune system. In addition to the similarity, zebrafish are completely transparent during early stages of development, and we can see single tumor cells establish under and grow into a tumor, so we can do live imaging of our zebrafish model to look at macrophage interactions with the tumor cells. By performing interspecies research analysis, I will identify evolutionarily conserved ways that Ewing sarcoma uses to resist standard therapies. By understanding these mechanisms, I'm able to pinpoint specific treatment targets applicable to humans, ultimately improving outcomes for these children.